Hello everyone, here I'm going to discuss about cofactors and enzymes, a very important concept as well as very interesting concept too. Okay, so first we draw the simple structure of enzyme. That's what we draw actually. Enzyme doesn't look like this, it's very complex. Okay, it looks like a bean shaped structure, but it is not bean. This particular enzyme always binds to the substrate, right? The substrate is looks like a block here. Okay, it's only for our convenient we draw such diagrams. Okay, so substrate is a chemical which normally binds to the active site of the enzyme, and ultimately enzyme substrate complex is produced, which looks like this. Correct. Okay, when substrate binds to the active site of the enzyme, and that complex is called as ES complex, enzyme substrate complex, and ultimately the enzyme brings conformational changes in the substrate by breaking the bonds of the substrate to release the products. You could see these tiny dots are products. Okay, we write it as P, and now this particular enzyme becomes free so that it can again bind to another substrate and then do the same kind of chemical reaction, that is catalyze the reaction. But actually, let me just tell you here, when enzyme binds to the substrate means enzyme should be in active state. Correct, it should be in active state, but there are many cases where enzyme is not active all the time. So this is very important. Okay, there are cases, okay, we write this word, cases where the enzymes are not active. So E, that is enzymes are not active all the time. Okay, all the time. Oh, my handwriting is going worst. Okay, so and let me just tell you that why it is not active all the time. Let us just see the answer for it. We'll find the answer for it. Let me just provide some conditions to this particular enzyme. Okay, let us give a name to this enzyme. Okay, we will name it as E itself. Okay, so this enzyme is called as E. Okay, so I'm going to provide certain conditions to this enzyme. Conditions to this enzyme now. Okay, condition number 1, let me provide a very good optimum temperature. Okay, so I'm going to provide optimum temperature to this enzyme. Okay, so we'll put a tick mark here, done. And then I'm going to provide one more condition, that is optimum pH. Okay, so tick. Next one, let me just provide one more thing, that is will remove or pull out all the inhibitors. Okay, so nobody is disturbing the enzyme now. Okay, so I'm going to remove all the inhibitors. So there are 3 different types of inhibitions or inhibitors are there, which always actually disturbs the enzyme and makes the enzyme kind of inactive, or which actually inhibits the production of products. Okay, so we are not talking about it. That means we just write some inhibitors are competitive inhibitors, sometimes non-competitive inhibitors will be there, or sometimes uncompetitive inhibitors will be there. Okay, so I have removed everything, so we'll put a tick mark here. So everything is now facilitating this particular enzyme to bind to the substrate and catalyze the reaction, but still the enzyme is not binding to the substrate. That means still enzyme is catalytically inactive. Okay, so we write that word here. After providing all the very good facilities, this enzyme is still catalytically inactive. Why this is catalytically inactive? Why? So that means there is some issues out there. What is that issue? The issue is very simple actually. The enzyme will work only when someone special actually comes and binds to the active site of the enzyme and makes the enzyme catalytically active. So we need someone special actually, a friend, good friend of this enzyme, to bind to the active site. Okay, so that only the enzyme becomes active. Got it? Okay, so I have to make this enzyme catalytically active. How can I make this enzyme catalytically active? Okay, so let me just this is the active site, right? Okay, so I'm going to bring a special person here. So can you see this, right? Okay, which now binds to the active site. Okay, so now once this person, or like someone, binds to the active site of this enzyme, the enzyme is now catalytically active, so it can actually bind to the substrate and ultimately produces products. Okay, so here we need to understand what is this, what is the name of this someone. The name of that someone is cofactors. Okay, so we write that cofactors. Okay, so what is this term cofactors mean? Okay, we'll just talk about that later. Now, before that, I just want to give you the structure of this enzyme. So normally enzyme is normally made up of a single polypeptide chain, correct? Okay, or sometimes you could see more than one polypeptide chain. Sometimes it could be like 2 3 polypeptide chains. Okay, normally enzyme is like this, and ultimately let me just crush it. Okay, because these polypeptide chains are coiled to form a complex structure. Now this is enzyme, see, got it? So this is actually the enzyme, and you could see a pocket like structure here. This is the active site of the enzyme. This pocket is active site. Okay, to this pocket, that is active site, someone, the friend, the cofactor, now this is cofactor actually, the someone, okay, which should bind. Now this enzyme is catalytically active. Yes, see, it's ready now. Okay, catalytically active. Now this is actually the substrate. Now the substrate can easily bind to the active site of the enzyme, and now the enzyme can catalyze the reaction. This is how the enzyme work. Got it? Okay, so it's not like simply you this thing, what you call a lock, can be opened by this particular key. Okay, it's not like that. So this lock is enzyme and this key is substrate. No, to open this I just have to supply a special person, that is nothing but this is cofactor. The cofactor should bind to the active site, then the substrate will bind to the active site. Okay, this is how the enzyme works. Okay, so I'm just activating this enzyme catalytically. Okay, so we see the thing here. Let me just give you the complex structure of the enzyme. Okay, this is the enzyme. It's made up of many polypeptide chain, or sometimes single polypeptide chain too. Okay, so let us assume that this is now enzyme, and you could see this is the active site, and to the active site the cofactor. The cofactor, I'll tell you the definition of cofactor, don't worry. Okay, so this is the cofactor actually. The cofactor now binds to the active site of the enzyme. Now this enzyme is activated, done. So it's activated, got it? Okay, it is ready to bind to the substrate and catalyze the reaction. Now we know that enzyme is made up of nothing but proteins, right? But simple protein, or like simply a protein, cannot actually bind to the substrate. It needs the cofactor. So the cofactor and enzyme togetherly makes a structure. Now this is actually called as holoenzyme. Okay, what is this holoenzyme? Holoenzyme is nothing but the protein part of the enzyme binds to the non-protein part, that is nothing but the cofactor. Got it? Now I have to tell you one more thing. When this particular enzyme binds to the cofactor, what do you call for this protein part now? The red color portion, that is the protein part. The protein part is called as apoenzyme. What do you call apoenzyme? What is apoenzyme? Apoenzyme is nothing but protein part of the enzyme. Okay, protein part of the enzyme, and we use this term only when this particular part of the enzyme binds to the cofactor. Otherwise I cannot call it as apoenzyme. Remember it. Okay, so apoenzyme is nothing but the protein part of the enzyme, and when it binds to the cofactor. So what is this cofactor now? The cofactor, this one, the green color one. Okay, cofactor is nothing but non-protein part of the enzyme. Non-protein part of the enzyme. So what is actually the mechanism of this non-protein part, that is cofactor? Mainly, it activates the enzyme catalytically, and now apoenzyme plus non-protein part, that is cofactor, is equal to holoenzyme. That's it. Very simple, right? What is holoenzyme? These two structures are nothing but called as holoenzyme, where you could see apoenzyme plus non-protein protein factor, that is nothing but called as cofactor. That's it. Okay, so this is very simple actually, so learn it. It's very simple. Let me just erase it, and let me just discuss the different types of cofactors. Okay, now we will see the first type of cofactor. There are three types actually. The first type of cofactor is nothing but that is called as prosthetic group. Prosthetic group. Okay, so what is this prosthetic group actually? The second type is called as coenzyme, and the third type of cofactor is called as metal ions. Okay, we don't have space here, tiny space. So let me just discuss about the prosthetic group. What is this prosthetic group? Very simple. They are organic compounds. Okay, What are they? They are organic compounds. Okay, so they are organic compounds. They are always found in association. Okay, oh sorry, association with the active site of the enzyme. Okay, so this is the thing to remember. Okay, the prosthetic group is nothing but enzyme. It's organic compounds, actually a type of organic compounds which is always found like bound to the active site of the enzyme. That means actually it's nothing but called as holoenzyme. So enzyme is always holoenzyme. That is nothing but apoenzyme plus the cofactor makes it holoenzyme all the time. Got it? Okay, so let me just give you one simple example, that is this one. Okay, so we'll bring this. So this is now the enzyme. This enzyme is active but catalytically inactive now. So to make it catalytically active, I have to bring this. So this is nothing but the cofactor. Got it? So let me just put this here. So cofactor binds to the active site all the time. Then it is called as prosthetic group. It binds to the active site of the enzyme all the time. Okay, and now this particular substrate can bind. Okay, I will put it here like this. Done. See, this is now enzyme substrate complex. Okay, for example, if I just remove this cofactor, this substrate cannot bind because enzyme is catalytically inactive. That's it. So we'll make it active again. So by just providing this cofactor. Okay, if this cofactor is bound to the active site of the enzyme all the time, then it is nothing but called as prosthetic group. That's it. Okay, it's nothing but organic compounds. Okay, it's non-protein part, organic compound. Got it? So what you call this? Cofactor, and this one is actually enzyme, the protein part. Now this is called as apoenzyme, and cofactor is equal to holoenzyme. This is holoenzyme. Got it? Now it makes a very good active enzyme now when it binds to substrate, and ultimately it produces the products. Okay, simple. Chalo. So we'll just bring it out, and let me give you a very good example. There are two examples for you. Example number one. So we just simply write one enzyme here. This is enzyme, and the red color thing. Okay, so this red color thing is actually the prosthetic group, which is already bound to the active site. It should be bound to the active site all the time. Okay, now it's this particular prosthetic group is nothing but heme. In this case it is heme, and the enzyme, the name of the enzyme is actually called as peroxidase. I'm giving a specific example. Okay, there is one enzyme which is called as peroxidase, and there is another prosthetic group that is nothing but a cofactor which is called as heme, which is bound to the active site. The red color one is nothing but heme here. Okay, so now this is the holoenzyme. Okay, the enzyme is catalytically active, and now we'll just I think I don't have any other color here, only three colors. Okay, so let us just bring the substrate for peroxidase enzyme is hydrogen peroxide. Okay, so we have H2O2 here. Okay, so ultimately this particular hydrogen peroxide will bind to this particular holoenzyme, that is peroxidase enzyme, and ultimately it's going to produce H2O plus O2. Got it? So this is the thing actually happens. And let me just tell you what is the fate of this enzyme now. After actually catalyzing the reaction, it's going to detach from the products, that is substrate actually, and but still you see in the active site of the enzyme, this red color thing is already like it's there actually. Okay, it is not detached, but the substrates, that is the products, are detached from the enzyme, but still the red color thing, the heme, is still there. The prosthetic group is still there. This is nothing but prosthetic group. That means the enzyme is always found in association with the prosthetic group or cofactor. Then it is called as prosthetic group all the time. Okay, that means this enzyme is catalytically active all the time. This is number one example. We go to the example number two. So we just write X2. So again I'm going to draw this enzyme now, and this thing is actually the heme group again. So this is heme group. So what is the name of the enzyme? This enzyme is a bit different actually. That is called as catalase. Second example, the enzyme is catalase. So here the prosthetic group is heme, which is bound to the active site. And now what are they going to do? So they are going to bind to this fellow, that is H2O2, and then they are going to produce the same product actually. Okay, so then after producing product, what is happening to the enzyme? Nothing. Okay, so see, this red thing fellow, the red fellow is still found with or associated with this enzyme. Got it? So holoenzyme. Got it? So this is actually prosthetic group. Hope you understood this. We go to the next, the second type. We'll erase this. That is actually called as the coenzyme. Okay. Right. Second type of cofactor is called as coenzyme. This is easy now. Now it's very easy because we all know that the cofactors, whatever the type of cofactor, doesn't matter, which always binds to the active site of the enzyme to make the enzyme catalytically active. Okay, so this particular coenzyme is nothing but, again, they are also organic compounds, that is similar to your prosthetic group organic compounds. But there is one small thing to say. This thing, the organic compounds, that is coenzyme, will bind to the active site of the enzyme, but only for a short duration or short time, that is transient. Got it? Okay, the organic compounds actually bound or bind to the active site of the enzyme, but it is only for a short time. That's it, transient. Got it? Okay. Why? Only when the enzyme is, okay, I think I should erase this. I'll write here. And the enzyme is bringing, or like, bringing a chemical reaction, that is catalysis. Okay, so when the reaction is going on, so this particular organic compounds, that is coenzyme, will bind to the enzyme, and now enzyme is active and ultimately reaction is completed. Got it? After the completion of the reaction, this particular coenzyme will detach from the active site of the enzyme. There is a bit of difference between prosthetic group and coenzyme, but it's very easy to understand actually. Okay, so we just will just give you one example, that is you are a bit familiar with this, NAD. What is NAD? Nicotine amide adenine dinucleotide. Okay, so NAD is nothing but a coenzyme, or even there is one more actually example, that is NADP. Okay, so this is also another type of coenzyme. What is common among NAD and NADP is, they have this vitamin B3, that is also called as niacin. They have niacin actually. Okay, so NAD and NADP are nothing but the coenzyme. They are going to bind to the active site of the enzyme only during a chemical reaction is occurring. Once the chemical reaction is completed, these particular coenzymes going to detach from the active site of the enzyme. Okay, I did not write. Okay, they are, oh yeah, I have mentioned it actually. They are going to bind to the active site. Okay, so next we'll go to the third type, that is, let me erase it. Okay, so we go to the third type, that is the metal ions. Okay, so we write metal ions. Okay, so metal ions, for example, like if you have seen any type of chemical reaction which is catalyzed by a particular enzyme, and when we write that reaction, we write something like Zn2 plus or Mg2 plus, correct? So we write these things. So this is nothing but metal ion. So they are very, very essential for a chemical reaction. Okay, so we see that. So for example, like this is the polypeptide chain of the enzyme. Okay, so now I cannot call this as apoenzyme. I can call this as apoenzyme when a particular cofactor binds to the active site of the enzyme. So now this is actually the holoenzyme. Now I can call this protein part of the enzyme as the apoenzyme, and this one is actually called as cofactor. Okay, so we have CF and apoenzyme, and ultimately holoenzyme is there here. We have holoenzyme here. Okay, so now let me just tell you, this cofactor in this case is the metal ion. Anything like zinc, for example, is easy to understand. Okay, zinc. but the thing is they are going to bind to the apo enzyme towards the ends of the side chains. Okay, for example, this is the polypeptide chain. Let us assume that the polypeptide chain is like this. Okay, so this is the end of the polypeptide chain, and particularly like a particular metal ion now binds to the side chains of the active site of this particular enzyme. So you could see here, this is actually the metal ion which is now attached or linked to the side chain, that is polypeptide chain of the active site of the enzyme. Got it? Okay, so here I just have to tell you that what type of linkage is there between the metal ion and the protein part of the enzyme. This is actually called as coordination bonds. Okay, so let me write here coordination bonds. Okay, so this particular metal ion links to these particular side chains of the active sites of the enzyme through coordination bonds. What is coordination bond? They are nothing but simple covalent bonds. Okay, I don't understand why these people make the words complicated. Okay, covalent bonds, simple. Coordination bonds are nothing but covalent bonds actually. So I'll give you a good example. Now this is holoenzyme, correct? Okay, so now what happens is, now let me just write this is substrate. Let me just put or make it a very complex because it's also chemical, right? Okay, so this is substrate, yes, and even it has got side chain, and this particular metal ion, when it is bound to the side chains of the active sites of the enzyme, okay, it is also binds to the side chain of the substrate, again through the same bonds, coordination bonds. Got it? Okay, now this structure is nothing but called as enzyme substrate complex. This enzyme is catalytically active. Done? Okay, this is how the things work actually. Okay, this is actually complicated diagram, but we can just draw a simple diagram. Okay, so this is simple. Should put green color here. So this is the metal ion and this one is the substrate. Okay, but you don't see the bonds actually. Got it? So these metal ions actually make bonds with that is coordination bonds with the enzyme as well as with the substrate. Got it? This is how actually they bring enzyme substrate complex, and enzyme is active and they are going to catalyze the reaction. Okay, so we just give you good example for this. Let me erase this. Example is in humans, that is during digestion process, we have one enzyme, that is carboxypeptidase. So to activate this enzyme carboxypeptidase, we need to supply a metal ion that is nothing but zinc. Okay, sorry, we just write zinc. We need to supply zinc. Got it? And then it will bind to the substrate, and then the enzyme will bring the reaction, that is actually catalyze the reaction. So that's all about the cofactors and enzymes. I hope that the concept is very clear now. Bit lengthy session, but I hope that it's clear. Thank you very much.